Now, when you look at the epithelial linings, whether you're talking the skin, epithelial lining of our lungs, our airways, or our gut epithelium lining basically from the stomach and the gastrointestinal tract. What we are seeing is tight columnar epithelium here and the stratum basal. They flatten out as you go to the stratum spinosum, granulosum, lucidium, corneum, and here in the skin, as they are replaced underneath from the stratum basal, from the stem cells here, and new layers are made, the older layers push out. They become dead keratinized cells. Keratin is a protein. All the interior of the cell is replaced by protein. So our outer skin cells are going to lack any organelles. They're dead for all intents and purposes. Tight bonds here at the bottom, watertight. As you get out, the bonds between the cells lessens, so you end up with lipids filling in the gaps. So you end up with a lipid layer here to keep fluid in, fluid out, and with the fluid out, also any microbes in the fluid. Underlying cells here that are still alive have those lamellar bodies, those granules of proteins, the defensin proteins. They will begin releasing. So as you look up at the close-up here, the lamellar bodies were still within the living cells, will begin releasing, flooding this lipid layer between the dead keratinized cells with these defensin proteins. So as these cells mature out, go out to the stratum corneum and the dead skin layers, they're also bringing along with them the defensin proteins, which will be there to break down, become activated, and begin punching holes in any microbes that may adhere to us. On the flip side, in our lungs, it's going to be a bit different. Sinuses in our lungs. We do not want to have dry, dead cells here acting as our barrier. We want to have nice living cells because we need gas exchange. We need to exchange gases. We also need to exchange up here in the sinuses temperature. We need to heat or cool off anything coming in. So we are going to have the columnar epithelium, and interspaced within the columnar epithelium, you're going to have goblet cells. Goblet cells are there to make this mucus you see here. Mucus lines everything. Bacteria going to adhere to the columnar epithelium, come into contact with the mucus. Most of them will get caught up here at the interface, and then as the mucus drains down the throat into the stomach, they get digested away. Some of them are able to overcome the mucus and then can adhere. Cilia, lining the lungs, lining the trachea. The ciliary escalator will pull stuff up from the lungs. Every time you breathe out, you breathe up some of the mucus with dust, bacteria, viruses caught in it, and you swallow it without even thinking about it. Underneath this epithelium, you're going to have a highly vascularized area. Highly vascularized areas that are bringing in blood, bringing in and out blood. You're going to have lots of ducts, mucus glands that are keeping this all moist. Keeping it moist with the mucus. Mucus has within it surfactants to help break down the surface tension so that the mucus flows. And as it flows, it's coming up out of the alveoli to the bronchioles, bronchii, up to the trachea, and down to the stomach. Side note, the reason why many people with cystic fibrosis have such issues is because these mucus glands produce the mucus, the goblet cells produce the mucus as they should, but the secretions are missing certain components. The surfactants, some of them being like chloride ions that should help thin out the mucus, are not there. So the mucus that's being produced by the goblet cells, produced by the mucus glands, is really thick. Since it's thick, it's trapping everything, but it's also itself getting trapped down in the alveoli. The ciliary escalator is not powerful enough to pull it up. So, cystic fibrosis patients, CF patients, end up with literally a version of pneumonia constantly. They cannot get the mucus up. It collects until it gets to a certain point they can't breathe. Emergency room visit where they will literally put a tube down there and will suck it out. So literally it's like a vacuum tube they put down into their lungs to suck it out so that they can resume breathing and they can reoxygenate their blood. In the gut epithelium, again, goblet cells making mucus. Here the mucus is not only acting as a barrier, but it is also acting as a lubricant. Got to keep pushing that food along. Can't have it get stuck in the microvilli. The tube that makes up the small intestine and large intestine, lined by columnar epithelium, interspersed with goblet cells and M crypts, the thing we see here as crypts. The crypts are areas that we are going to have cells designed just for antimicrobial activity. Notice here the artist, when they drew this crypt here, they did not draw in microvilli, and there is a reason. The crypts are just a pocket, a small pocket. It's not a groove as it's showing here. Remember, this is going to be in 3 dimensions coming at you. So this is like a little indentation, a small indentation, only a few cells wide. Well, lacking the microvilli means that these cells are not there for absorption. They are here to produce antimicrobial proteins, defensins and Reg3. Defensins Reg3 will begin poking hole in microbes. Yes, I know we want to have some microbes there, but not a lot of them. We want to keep these numbers in check. They overgrow. It could cause problems for you later. So these Paneth cells they're showing here at the base of the crypt produce the defensin proteins, which will then flood into the small intestine. Keeps these numbers in check. You want some, just not all.